The fourth lecture in this series basically has to do with a very unique and special bird, the bar-headed goose. How does the bar-headed goose? This is a puzzle that has been for many years. People have questioned how it was possible. Bar-headed geese are supposed to be the paradigm of high-altitude flight. They migrate over the Himalayan mountain range. From their breeding grounds in Mongolia, they go down to the southern India. Of course, we know that flight is energetically expensive. It's expensive to fly. So flying at high altitude must be even more expensive, because not only you do it at high altitude, there is less oxygen, and it's much colder. The question has been, yes, there's been signs of these birds flying at this altitude. But can we characterize this? Interestingly, there's a relatively recent paper published in the Proceedings of the Royal Society B, The Paradox of Extreme High Altitude Migration in Bar-Headed Geese and Cerindicus. And what these authors have done is instrument these animals with collars, with GPS tracking systems, to know what they are doing and how they are doing it. And this is the basic picture. The basic picture is that from the breeding grounds in Mongolia and China, these animals will migrate over the Tibetan Plateau down to their overwintering grounds in southern India. The interesting thing is that if you take this and you chart this in a map from their breeding grounds to the place they end up, the straight line, the shortest route actually is the white line. The shortest line is the white line, and notice that the shortest line passes through these white crosses. Each of these white crosses indicates one 8,000 meter peak. You know that there are 14 in the Himalayan and Karakoram range here. This is the straightest line. This is the fastest, or this is the shortest distance. When you trace the routes that the animals take, a bit on the side, 112 kilometers longer. The animals take a route that is not the shortest one. It's about 112 kilometers longer. Why is that so? Well, on the one side, if they cross the Himalaya over this area here, this is actually the lowest part of the Himalayan range they can cross. On the other hand, notice that by taking the plateau or attacking the plateau further down here, they are also a shorter period of time at the high altitude. And that's what you see in the next range. This is the Tibetan Plateau, from the plains in India and the plains in Mongolia. This is the high altitude reaching here. So the relatively little data that we have available is suggesting that these animals do not take the straightest route and may not go the longest time at the highest altitude. This is actually a rather complex picture, more like on the, well, let's see. These are, in red, you have areas over 5,000 meters. And this picture is actually a part of one of these squares, this or the next square. From the looks of it, this is the first square and this is the second square. The same thing here, the same thing here. These are essentially the only difference between these three pictures is that in this you have in red areas above 5,000 meters. In this one, areas above 5,500, and in these areas above 6,000 meters. So, if you now zoom in into these pictures, a black dot is the position of a bird. A black dot in a red area suggests birds that are flying at that altitude. But notice that that does not happen so often. Most black dots are actually on white areas, meaning that are areas, in this case, below 5,000 meters, or in this case, below 5,500 meters, or in this case, below 6,000 meters. Pretty much no black dot is on a red spot here. The conclusion is that bar-headed geese rarely migrate above 6,000 meters. People have seen them above, fine, but they rarely, when you look at the picture, the data we have, they don't migrate so high. The maximal flight altitude that has been measured in these birds is actually 7,300 meters for the southbound flight and 6,500 meters for the northbound. I say just. I think that is quite a feat. I think it's impressive that a bird can do this. But it's certainly less of the impression that people were starting to, or that we were believing, that they were flying over the Himalayas. Mm-hmm. But they still can achieve this. It's quite an impressive achievement. Mm-hmm. What mechanisms make the bar-headed goose then different from other duck species? One duck species that has been compared with extensively is the Peking duck that you have here. This is the bar-headed goose. The mechanisms that make them different are the following. A strong hyperventilatory response to poikilocapnic hypoxia. So these are animals that do not seem to care that much about the low hypocapnia, do not seem to care much about the fact that when you hyperventilate, CO2 becomes low. They still show a strong ventilatory response. They have an increased affinity of hemoglobin for oxygen. They have an absence of hypoxic polycythemia. And this is a recurrent topic that we will see also later in the adaptation of human populations. In bar-headed geese, there is no increase in hemoglobin with altitude. They do not produce more red blood cells. They do not produce more hemoglobin. There is also high capillary density in flight muscles. They redistribute the mitochondria so that they reduce oxygen diffusion distances. You will see that in a second. The mitochondria are placed under the plasma membrane instead of inside the cell. They have a blunted hypoxic pulmonary vasoconstriction. These animals show less of a vasoconstrictor response. And finally, they have hypocapnic brain vessel insensitivity. Their brain blood vessels are insensitive to hypocapnia. This is not a feature specific for the bar-headed goose. It actually seems to be present in all birds. Birds seem to have an insensitivity to hypocapnic brain vessels. This is the cause of high-altitude cerebral edema in mammals, in humans. But it seems by default, birds do not have this sensitivity, therefore could not get high-altitude cerebral edema. Let's look at one at a time. The strong ventilatory response, the strong hyperventilatory response. If you compare the bar-headed goose up here with other geese species or even ducks, what you see is that for an inspired partial pressure, percentage of oxygen, the bar-headed goose always has a higher arterial PO2. That means that even if you go hypoxic, the bar-headed goose has more oxygen in the blood than the other species. Eventually, what you see is that the bar-headed goose can be pushed down to 4% of oxygen. The other ducks will not make it. And this is actually the result, or this happens because at the low, or in hypoxic conditions, at low fractions of oxygen, the bar-headed goose hyperventilates much more. Notice the extreme hyperventilation in comparison with the other two species, almost double, you could say. So the bar-headed geese is hyperventilating extensively. Is there a problem? And then what you see here is that this hyperventilatory response is occurring only in poikilocapnic conditions. Only in poikilocapnic conditions. These are experiments done in the lab where they can actually control the level of CO2. If you let the animals go poikilocapnic because they are hyperventilating, the response is maximized. If the animals are isocapnic, meaning that you control the levels of CO2 and you reduce them in the lab, there is no difference between the bar-headed goose and the Peking duck. Another response that I emphasize, the absence of hypoxic polycythemia. There is no increase in hematocrit or hemoglobin concentration. The question and the guess is that this facilitates cardiac function in hypoxic conditions because an increase in hematocrit will increase the viscosity of the blood and will impose a larger cardiac work. This is actually something that you can see in this picture. These are animals flying in a wind tunnel, and when you fly barnacle goose or bar-headed goose in this case, what you see is that the heart rates in relation with oxygen consumption, at the same heart rates of 400 to 500 beats per minute, the bar-headed goose has a much higher oxygen consumption. These animals seem to deal better at the same heart rate, are able to extract more oxygen from the circulation from the blood. They also have an increased affinity of hemoglobin for oxygen. So their hemoglobin has a higher affinity. This is the hemoglobin oxygen dissociation curve for bat-headed goose. The P50 is lower, they have a higher affinity. And this is because of changes, mutations in the hemoglobin primary structure. This higher affinity partly compensates for the absence of polycythemia. So what these birds are doing, they are not increasing hemoglobin concentration. Instead, they are reducing affinity. And they are increasing affinity by reducing P50. And that actually partly compensates for it. They also have important structural differences in flight muscles, high capillary density and redistribution of mitochondria. This has been shown in this paper, as you see in these pictures. On one side, higher capillarization. The capillarization in the bar-headed goose, these bars, in muscle, is higher. And that's what you see here. The capillaries are these darker dots among the muscle fibers. Increased capillarization. At the same time, in comparison with the other goose species, what you see is an accumulation of mitochondria. These gray dots are accumulation of mitochondria under the plasma membrane. This would be the outline of the muscle fiber. And notice that the mitochondria tend to accumulate under the sarcolemma. Why? This is where the blood is actually perfused. This is where the capillaries are. Therefore, you are reducing the diffusion distances. If you have a mitochondria in the middle of the fiber, or if you have a mitochondria here, obviously the oxygen will reach the mitochondria if it's closer to the plasma surface. And that's actually what's happening and what's shown by this distribution of mitochondria in these species. This is actually the bar-headed goose. Finally, you have a differential blood reactivity. A smaller hypoxic pulmonary vasoconstriction. If we were saying that hypoxic pulmonary vasoconstriction is the main threat to a climber climbing at altitude because of the danger of high altitude pulmonary edema, in the case of the bar-headed goose, also in the case of yaks and llamas, there is a reduced hypoxic pulmonary vasoconstriction. How do you see this? You see this in this graph. As the oxygen inspired or the arterial oxygen partial pressure is lower, the mean pulmonary arterial pressure increases in all species. Increases much less in the bar-headed goose. That means that the bar-headed goose is much more tolerant to hypoxia. There is no hypoxic pulmonary vasoconstriction, or there is less hypoxic pulmonary vasoconstriction. Finally, in the other side, the cerebral circulation. There is no reduction in cerebral blood flow as a result of hyperventilatory hypocapnia. When the animal hyperventilates, partial pressure of CO2 goes down. That's what we call as hypocapnia. As a result of this lowering of CO2, cerebral blood vessels in mammals vasoconstrict. Bird cerebral blood vessels do not vasoconstrict. Therefore, you can maintain cerebral blood flow. This is, as I said, common to all birds. It's not specific for the bar-headed goose. So we cannot claim this to be an adaptation in the bar-headed goose, but it's apparent in all bird species. So, how would I summarize this? High altitude adaptations in the bar-headed goose. They are able, they can cope with a hypoxic high altitude environment, and that is because they have done multiple changes on the cardiorespiratory system, and including the blood. I would say that the bar-headed goose is a good example of acclimatization changes, if you want to put it this way. It may be a bit early to claim them as being adaptations, but at the same time, we know that the bar-headed goose has been doing this for thousands and thousands of years. The problem is that we have, as it is at the moment, no reconstruction of all these adaptations in a historical context. And that may require a little bit more results. despite all these responses, which are beneficial at altitude, measurements done of cardiac output in hypoxic conditions suggest that the bar-headed geese could not maintain powered aerobic flight to fly over Mount Everest. But that is powered aerobic flight because it's costly. Remember that many birds that fly very high, vultures for example, do so by gliding. And gliding does not require the same amount of energy because it does not require muscles. It's basically extending the wings, and on the surface of the wings these animals are able to move using thermal currents, etc. But the estimations that have been made suggest that a bar-headed goose could not fly by flapping the wings over Mount Everest. Of course, it's up to the bar-headed geese to prove us wrong, and I'm sure that they should be able to do that. But these are what our estimations say today. I want to remind you that in 1980, the Everest expedition predicted, based on the measurements they did, predicted that it was impossible to climb Mount Everest for a human without supplementary oxygen. Not many years later, a couple of years later, Reinhold Messner did it without supplementary oxygen. So, even for us, predictions have not been maintained, and we are much more capable than what physiologists may believe. But this is at least the data we have on cardiac output in these birds at the moment. That means that there's always more data that could be helpful. I would encourage you to watch this YouTube film. It's a very short film, about a minute, of a goose flying in a wind tunnel. And basically you'll see Jessica Meyer there, just flying this bird, a beautiful demonstration of what these eyes can do and what we can do with them in research-wise. Thank you very much.